Okay, now let's move on to question number 6. Particulate matter is solid particles and liquid particles suspended in air. Polluted air contains more particulate matter than clear air. A high concentration of particulate matter results in the death of some alveolar epithelial cells. So obviously if there's more pollution, there's more particulate matter in the environment, so that will damage our alveolar epithelial cells. Let's continue reading. If alveolar epithelial cells die inside the human body, they are replaced by non-specialized thickened cells. And because they are thick cells, they will create problem in the exchange of the gases. Explain why death of alveolar epithelial cells reduces gas exchange. That is for 3 marks. Here we have the alveolus, consists of a single layer of cells, and this is how the exchange of the gases between the alveolus and the blood takes place. So the answer should include number 1, it reduces the surface area. There are no more alveoli, they have died out. So that's surface area. And number 2 increases the distance of diffusion. And remember we said they will be replaced by thick tissue, so that increases the distance of diffusion. So there will be problem in diffusion of the gases. And number 3, it reduces the rate of gas exchange. The next part of this question is scientists grew alveolar epithelial cells and exposed the epithelial cells to different concentrations of particulate matter. They calculated the percentage of these alveolar epithelial cells that died after 24 hours of exposure to our particulate matter. The results are shown in the figure 6. So here we can see the concentration of the particulate matter, so 0, 5 and 100 per microgram per cubic centimeter. And on this side we have the percentage of dead cells after 24 hours of exposure. So obviously as you can see, if the concentration of the particulate matter is less, then there is a less death of the cells, and as you increase the concentration of the particulate matter, the percentage of dead cells increases. Now the question is, do the data in figure 6 show a linear relationship between concentration of particulate matter and percentage of dead cells? Use suitable calculations to justify your answer. So one thing is very clear, that as we increase the concentration of the particulate matter, it increases the percentage of the dead cell. But is that a linear relationship or not? If that's a linear relationship, then we will see the same trend. For example, we will see the same trend in the slope. If the slope or gradient between 0 and 5 and 5 and 100 is the same, so that means there is a linear relationship. Okay, so slope between 0 and 5 would be something like that. To find the slope is really simple. All you need is the vertical distance and the horizontal distance. Okay, so the vertical distance would be the distance from here to here. But for that we need to know how much each small scale would be. So the distance between here to here is 15, and that is 10 small squares. So 15 divided by 10 would be 1.5. So that means each small square is equal to 1.5. Okay, so the slope from 0 to 5 will look something like this. Let's say we call it m1. To calculate the slope, we will need the vertical distance and the horizontal distance. The vertical distance would be from here to here, and the horizontal distance would be from here to here, which is equal to 5, because the distance from here to here is 5. Okay, so the vertical distance is equal to 6 small squares, and because each small square is equal to 1.5, so the total distance would be 1.5 times 6, and that would be equal to 9. So slope would be vertical distance divided by horizontal distance, and that would be equal to 1.8. Okay, so now we calculate the slope from 5 to 100. And that would be from here to here. Let's say we call it m2. And again, that would be equal to vertical distance divided by horizontal distance. So the vertical distance, if you calculate, that will be 27. And the horizontal distance is from 5 to 100, and there would be 95. So slope in this case would be vertical distance 27 divided by 95 horizontal distance, and that would be 0.28. Now we can clearly see that both these slopes are not the same. Therefore, we can say that there is no linear relationship between the concentration of the particulate matter and the percentage of the dead cell. Question number 7. Alpha-gal is a disaccharide found in red meat. Alpha-gal is made of 2 galactose molecules. Galactose has a chemical formula C6H12O6. Give the chemical formula of the disaccharide alpha-gal and describe how it is formed from 2 galactose molecules. So that question is for 2 marks. So you have to give the formula of alpha-gal and a little description. It's a pretty simple question. So all we need is a formula for alpha-gal, which is composed of 2 molecules of galactose. And we have also been given the formula of the galactose. So alpha-gal is a disaccharide which is made of 2 monosaccharides of galactose, and this is how the alpha-gal looks like. So this is one galactose molecule and its formula is C6H12O6. C6H12O6, and similarly we have here second galactose molecule, and again its formula is C6H12O6. If you remember, the reaction between two monosaccharides is a condensation reaction. That means a water molecule is released when two molecules combine. So this is where the condensation takes place and a water molecule is removed. So if you look carefully, we had OH here, which is no more here. So OH from here and hydrogen from here, and a bond is formed between the two molecules. So if you talk about the formula of the alpha gal, so that would be C6H12O6 plus C6H12O6 minus H2O. So the formula would be C12H22 and O11. And in the description, we will say this is a condensation reaction. Okay, the next part of this question is, some people eat red meat for many years without having a reaction, then have an allergic reaction to the alpha gal in red meat. An allergic reaction is caused by an immune response. Draw a level diagram of an antibody and identify the specific alpha gal binding site. Okay, so it is asking to draw a label diagram of an antibody and identify the specific alpha gal binding site, or in general the binding site of an antibody, which is same for all the antibodies. So this is how the antibody looks like. It's a Y-shaped structure having a heavy chain and a light chain. The top part of both the heavy and light chain is called the variable region, and this is where the antibodies differ from each other. The rest of the bottom part is the constant region. So the binding of the antigen takes place here at the end. So that's why it's called the antigen binding site. And that would be the same for the alpha gal as well. Okay, so the next part of the question is, a tick is a small animal that bites humans and feeds on their blood. This results in the proteins from the tick saliva entering the human body. Scientists have suggested one hypothesis for the allergic reactions to alpha gal in red meat. They think that an earlier immune response to a tick bite can cause a person to have an allergic reaction to alpha gal in red meat. Suggest how one antibody can be specific to tick protein and to alpha gal. And that question is for 2 marks. So basically, alpha gal and the protein in the tick saliva have very similar structure. Therefore, antibody is complementary to both these antigens. So your answer should include these points. Number 1. Part of the tick protein and alpha gal have similar structure or shape. Number 2. Antibody is complementary to both tick protein and alpha gal. Or you can say antigen binding site is complementary to both tick protein and alpha gal. Or you can say antibody can form antigen-antibody complexes with both tick protein and alpha gal. The next part of the question is, scientists took blood samples from one man over several weeks and measured the concentration of antibody in man's blood. During this time, the man had 2 tick bites and had an allergic reaction to alpha gal in red meat. The results are shown in figure 7. The scientists' hypothesis was that an earlier immune response to tick protein causes the allergic reaction. Consider whether figure 7 supports this hypothesis. Okay, so what you see here is that we have the antibody concentration over various period of time. So the solid line indicates the monoclonal antibodies specific to alpha gal, which is this one, while the dotted line represents the total antibody. So these antibodies are just specific to alpha gal, while this antibody titer is overall antibody titer in the body, which can be for hundreds or thousands of antigens. And then you can see here 2 arrows, one here and one here. So this indicates the first bite of the tick, while the second arrow indicates the second bite of the tick. So as you can see here, after the first bite, the antibodies against the alpha gal started to increase. After the second tick bite, the antibody levels started to increase rapidly. This is because they are the memory cells after the first exposure. There is the memory cells. So if there's a second exposure, the antibody response is much faster and much quicker and much higher. And then after a certain period of time, the response declines. This is clear because this is the response against the alpha gal. However, we see here that the total antibodies are also increasing. So that means it's not just the antibodies against the alpha gal which is causing the allergic reaction, but there are other antibodies as well which are contributing to allergic reaction. So your answer should include all these points. Number 1, exposure to tick protein is followed by increase in antibody specific to alpha gal. Makes sense. Number 2, later, greater or faster increase in antibodies suggests that there are memory cells, which we just talked about. And then number 3, antibodies specific to alpha gal increases during or after allergic reaction. And number 4, during or after allergic reaction, total antibody increases more than alpha gal antibody, which we can see here in the dotted line. So maybe other antibodies that are causing allergic reaction. So that means it's possibly the other antibody which are causing the allergic reaction because their titer is much higher compared to the alpha gal. Question 8. The first part of the question is complete table 2 to show 3 differences between DNA in the nucleus of a plant cell and DNA in a prokaryotic cell. So it's a simple question. It's simply asking you the difference between the eukaryotic, because plants are eukaryotes, and the prokaryotic DNA. For example, prokaryote include bacteria. So all you have to do is just tell the difference between a normal eukaryote cell and the bacteria cell. So let's have a look at the comparison of both these DNA side by side. So on the left side, we have the DNA from the plants or the eukaryotes. So the first difference is that DNA is linear, while in case of bacteria, the DNA is not linear, but it is circular. As you can see here, it's all circular. Second, in the eukaryotic DNA, you will see a lot of histones, as you can see here. These are the proteins associated with the DNA, while in case of the bacteria, you won't see any histones, but it's just the DNA. The third difference is that in case of prokaryotes, you will see many plasmids, while in case of eukaryotes, you won't see any plasmids. And eukaryotic DNA is much longer compared to the bacterial DNA. So you will include any 3 of these points. Number 1, associated with histones proteins versus no histones proteins. So histone proteins in eukaryotes and no histone proteins in prokaryotes. Number 2, eukaryotic linear, prokaryotic circular. Number 3, no plasmid in eukaryotes, plasmids in prokaryotes. Number 4, introns. If you look at the DNA, you will see a lot of introns in the DNA. So introns are basically these, which is a non-functional DNA, but the exon is a functional DNA. So eukaryotic DNA has a lot of introns, while in case of the bacteria, you won't find any introns. The next part of the question is scientists investigated the genetic diversity between several species of sweet potato. They studied non-coding multiple repeats of base sequences. Define non-coding base sequences and describe where the non-coding multiple repeats are positioned in the genome. So that question is for 2 marks, and this is basically what we have just done in the last part. So all it's asking is about the non-coding DNA, which is also called the introns. So as we have just seen, introns are the sequences of the DNA which do not code for any protein. There are many introns in the eukaryotic DNA. So your answer should include number 1, DNA that does not code for protein or polypeptides, or you can say DNA that does not code for sequences of amino acids, or you can say DNA that does not code for transfer RNA or ribosomal RNA. And the second point would be its position between genes. As you can see here, so introns is between the genes. So exons are the working genes, introns are in between them. The next part of the question is the percentage similarities in the non-coding multiple repeats of base sequences of 4 species of sweet potato are shown in table 3. And the question is, use the information in the table 3 to complete the phylogenetic tree shown in figure 8 and write the letter that represents the correct species into each box. So we have to write here the correct letters in these boxes. So basically in this table, we see there are 4 different species of sweet potato, and they have been coded as C, L, R, and T. And here on this side, we have the percentage similarity between non-coding multiple repeat base sequences. So this is basically just the similarity between the DNA sequences. So the more the similarity between the species is, the more related they are, and the closer we will draw them on the phylogenetic tree. So if you look here, so the highest similarity is here, 59.7, and that is between C and T. Or you can see even here 59.7 between T and C. So if you look at the phylogenetic tree, so these two are very closely related. So obviously one of them has to be C and one of them has to be T. It doesn't matter, you can write first C or T. Now we have 2 species left that should fill in here and here. Obviously the one goes here is the one which is more similar to C and T, and the one which goes here will be the one which is least similar. So if you look at the similarities, the similarity between C and L is the next 53.5. So that means C and L are very closely related. Similarly, you can see here that similarity between T and L is also 53.7, which shows that T, C, and L are very closely related. So we already have C and T here. So that means the next would be L, which is very closely related to both C and T. And finally, we have R, and you can see R has the least similarity either with A or L. So it should be farthest away on the tree. So R should go here. The next part of the question says, the scientists studied 5 individuals from each species. Within the 5 individuals of the species T, they found a percentage similarity of 66%. Use table 3 to evaluate how this information affects the validity of the phylogenetic tree. Okay, so we just have seen that the maximum similarity is 59.7. But here it says that within species T, they found a percentage similarity of 66%, which is higher than any we have just seen. So what does it mean? So if you look carefully, it says the scientist studies the 5 individuals. So this is a very small number. So because it's a very small number, so it can't give you a representative value. So that could be the potential reason. So your answer should include, number 1, more similar than with any other species, because obviously we haven't seen 66% in any other species. Number 2, not spotted because high intraspecific variation in the species T compared with variation between T and C. And number 3, small sample size, or only because we have 5 individuals, so that could be the reason. Okay, now let's move on to question number 9. Figure 9 shows 3 examples of the square fields of view the scientists used to calculate a mean stomatal density. The question is, calculate the mean stomatal density in the 3 fields of view in figure 9 and give your answer as number of stomata per square millimeter. Okay, so what we see here, so we see here stomata. So now you see here the number of stomata in 3 different field views. So all these dots are actually representing the stomata. And if you look carefully, each represents 250 microns. So that means this is a 250 micron and this will also be 250 micron. Because we need to provide the answer in per square millimeter, so it would be good if you can convert this into millimeter. So 250 micrometer would be equal to 0.25 millimeter, because a 1000 micrometer is equal to 1 millimeter. So this length is 0.25 millimeter and this length will also be 0.25, and if you multiply this, so you get 0.25 times 0.25 and that will give you 0.0625 square millimeter. So the area of this square is 0.0625 square millimeter. Okay, so since we need to find the mean stomatal density, so first we need to find the mean or average of the stomata. So we need to count the number of stomata here. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So we have 11 here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So again we have 11 here. And 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 and 10 here. So the average would be 10 plus 11 plus 11 divided by 3, because we have 3 readings, and that would be 10.7. So that means 0.0625 square millimeter has 10.7 stomata, and in 1 square millimeter there would be 10.7 over 0.0625, about 171 stomata per square millimeter. The next part of the question is, the scientists used leaves from individual trees that had grown in the different areas of the world in different years. Each tree had grown in an area and year with known concentration of carbon dioxide. The results are shown in the figure 10. And the question is, give a null hypothesis for this investigation and name a statistical test that would be appropriate to test your null hypothesis. Okay, so first we need to understand what this figure is. So what we see here is the carbon dioxide concentration in the atmosphere, and that's in parts per million or ppm, but on this side we have stomatal density or number of stomata per square millimeter. So that means if the carbon dioxide concentration decreases as we move left, the number of stomata per square millimeter increases. This is because if there's less carbon dioxide in the environment, then the plants will tend to have more stomata so they can absorb more carbon dioxide. In other words, there is an inverse correlation between the carbon dioxide concentration and the number of stomata. Okay, now let's go to the question again. So the question was, give a null hypothesis for this investigation and name a statistical test that would be appropriate to test your null hypothesis. So obviously the null hypothesis would be that there is no correlation between the carbon dioxide concentration and stomatal density. So your answer would be, there is no association or correlation or relationship between the concentration of carbon dioxide and the stomatal density, or you can say the concentration of the carbon dioxide does not affect the stomatal density. And the second part was the statistical test that would be appropriate. Obviously we can't use here t-test because we don't have the mean values. We are not comparing the means. So we're just checking the correlation between the two values. So the appropriate test here would be the correlation coefficient. Okay, so the next part of the question says, from 1910 to 2000, the carbon dioxide concentration in the atmosphere increased from 300 parts per million to 365 parts per million. Use figure 10 to calculate the mean rate of change in stomatal density from 1910 to 2000 and give your answer as a number of stomata per square millimeter per 10-year period. Okay, so we need to find here the mean rate of change in the stomatal density, and we need to provide the answer as the number of stomata per square millimeter per 10-year period. Okay, so let's compile all the information that we have. So we have data from 1910. So in 1910, the carbon dioxide concentration was 300 ppm, and then in 2000, it increased to 365 ppm. So next we need to see this 300 ppm corresponds to what stomatal density. So if you look at the graph, this is 300, and if you move up, it comes to be here. So it comes here, which is 202. So the stomatal density was 202. Okay, so the next is 365 in year 2000. So this is 360, this is 370, so 360 will be right in the middle. So if you keep moving up, we get this value, which is 178. Therefore, the stomatal density decreased from 202 in 1910 to 178 per square millimeter in year 2000. And that is a difference of 24. So this means between 1910 and 2000, the stomatal density decreased by 24. And that's over a period of 90 years. Since you're supposed to give the answer in 10-year period time, 24 divided by 90 times 10, and that would give you 2.7 per square millimeter and per 10 year. The next part of the question is, a journalist saw figure 10 and suggested that future increases in atmospheric carbon dioxide concentration could result in less transpiration. Evaluate his suggestion. So that question is for 4 marks, so you need to provide a bit more of detail. So your answer should include these points. So the number one was be increasing the carbon dioxide concentration shows a decreased stomatal density, which we have just seen. So few stomata mean less transpiration. Few stomata, so there will be less chance that the water will be lost. Few stomata mean less evaporation of water. And the third point would be same volume of carbon dioxide can be absorbed for photosynthesis with a smaller number of stomata. The next point is, the problem is we do not know the size of the stomata. We do not know whether leaf size has changed. We do not know if this is true of all species of plants. We do not know how long the stomata are open for, and we do not know if this trend will continue beyond the concentration of carbon dioxide shown in figure. And other factors affect transpiration rate as well. So we can't say for sure that future increases in atmospheric carbon dioxide concentration could result in less transpiration. Okay, so now the question number 10. So the first part of the question is, describe how messenger RNA is formed by transcription in eukaryotes. That's a straightforward question, but it is for 5 marks, so we need to provide a bit of detail. So just to remind you how the transcription works. So the transcription is when DNA makes messenger RNA, which is actually a copy of the DNA. So this process of making messenger RNA from DNA is called the transcription. So this is basically pre-messenger RNA, which is then further processed to make messenger RNA, and messenger RNA then moves out of the nucleus, goes in the cytoplasm where it's translated, and that process is called translation. And during this process, polypeptide or proteins are made. So simply what happens with this process is that DNA, which is double-stranded, uncoils and separates its strands, exposing its nucleotides. And one of these strands is then used as a template, and nucleotides are joined together using RNA polymerase, which joins these nucleotides together to make the new messenger RNA. And once this process is completed, DNA comes back to its normal coil shape. So in the answer, you can say that number 1, the hydrogen bonds between DNA bases break down. Number 2, only one DNA strand acts as a template, as we have just seen. Not the 2 of the strands act as a template, but only one of them. Number 3, free RNA nucleotides aligned by complementary base pairing. Obviously, that's a complementary base pairing. That means C will always pair with G, and A will always pair with T, which in case of RNA is called uracil or U. Number 4, in RNA, uracil base pairs with adenine on DNA. Number 4, in RNA, uracil base pairs with adenine on DNA. Number 5, RNA polymerase joins adjacent RNA nucleotides. Number 6, by phosphodiester bonds between adjacent nucleotides. And number 7, pre-messenger RNA is spliced to form messenger RNA. So basically, the introns are removed during this process to make the final RNA. Okay, the next part of the question is, describe how a polypeptide is formed by translation of messenger RNA. Again, it's for 6 marks, so we need a bit of detail here. So basically what it's asking here is asking you about the details of the translation, which is when the messenger RNA goes from the nucleus into the cytoplasm and starts making proteins or polypeptides. So what happens during this process is messenger RNA, which has just come out of the nucleus into the cytoplasm, attaches itself to the ribosome. So messenger RNA has a specific sequences. 3 of these adjacent sequences is called a codon. So we have one codon here, we have another codon here, and we have another codon here. And then we have various transfer RNAs in these cytoplasms. Each of these transfer RNA carries a certain sequence which is complementary to the sequence of the codon. Each transfer RNA is carrying an amino acid. So all these transfer RNAs bind on the specific codons and bring an amino acid, and then a peptide bond is formed between the 2 amino acids. That process keeps going on and we get a long polypeptide chain at the end. So your answer should include these points. Number 1, messenger RNA attaches to ribosomes. Number 2, transfer RNA anticodons bind to complementary messenger RNA codons. Number 3, transfer RNA brings a specific amino acid. Number 4, amino acids joined by peptide bonds. Number 5, amino acids joined together with the use of ATP. Obviously, we need energy. That energy comes from the ATP. Number 6, transfer RNA released after amino acids joined to polypeptide. So once the transfer RNA has given its amino acid, it detaches itself. And number 7, the ribosome moves along the messenger RNA to form the polypeptide. Okay, the next part of the question is define gene mutation and explain how a gene mutation can have no effect on an animal and a positive effect on an animal. So we have to explain both the situation when it has no effect and when it has a positive effect on an individual. And that question is for 4 marks. Let's see what the mutation is and what effect can it have. So on the left hand side we have a normal gene, so it gets transcribed and translated into a normal protein. While on the right hand side we have a mutated gene, and here is a mutation. Now depending on the type of the mutation, it can have 2 different effects. Either it can still produce a protein, but that would be an abnormal protein, or it will produce no protein at all. So your answer should include these points. So number 1 will be the definition of the gene mutation, which is the change in the base or nucleotide sequence of the chromosome. Number 2 results in the formation of a new allele. So that is the definition. Now the first case when it has no effect, because genetic code is degenerate. So amino acid sequence may not change. Or we can say the mutation is in an intron, so amino acid sequences may not change, because introns are non-coding sequences. So if there's a mutation in the intron, that will have no effect because introns are not going to be translated. Number 4, does change amino acid but no effect on tertiary structure. So this means it did change the amino acid, but the overall structure of the protein was still the same, so protein was still functioning properly. Number 5, new allele is recessive, so it does not influence phenotype, because it was recessive, so it didn't have any effect. Okay, now the second situation when the mutation has a positive effect, that results in change in a polypeptide that positively changes the properties of the protein, or it results in the change in the polypeptide that positively changes a named protein. It may result in increased reproductive success, or it may result in increased survival chances, both of which are positive effects.